Okay, this is the Atheist Community of Austin's monthly lecture, and I was trying to find a speaker for today, and I kept not getting anybody, and then finally somebody said, well, I'd love to do it, but it's Mother's Day, and I said, oh, okay. So I said, well, my mother's dead, so I don't have to worry about it. So I can do it, and if nobody shows up, then I can practice my slideshow, and so you get to participate in my practice. All right, so the microbiome. I had a little piece here that some people listen to NPR on a regular basis, and there was a little clip of Jim is not the farthest one back. It is broken and precedent. Okay. Anyway, there's a little clip that it talks about. It is kind of an introduction to the concept of your microbiome. And the thing is, the terminology is sometimes a little confusing for folks, but there are two words that are related, the microbiota. Biota refers to living organisms, and so that refers to all the different kinds of organisms that live on. When they use the term microbiome, they're referring to the genetic makeup of all the organisms. So when you look at the total number of cells in a person, about 10% of them are actually human cells, and the other 90% are some kind of bacterium or protist or some other kind of thing. Now, since that represents several thousand different species of organism, when you look at the total number of genes, they're about 1% of the total gene content is human genes. And we've known this for a while, but what we haven't known until relatively recently is how those genes actually can affect us almost as much as our own human genes affect us. And we're going to see some examples of how that happens. It's an extremely broad topic, and it's relatively new, so there's a lot of stuff here. And I have taken some clips from different presentations. And if you're interested in following up, if you will send me an email, I have a file of links to other videos. One of them is about a 2-hour-long video. It has about 3 or 4 different scientists that have been working on different aspects of it, and they all kind of link together. And another one that's about a 30-minute video, which is also pretty interesting. And they are a little bit on the technical side, and that's part of the fact that they're long and technical is part of the reason that we're not going to see them today. So, your microbiome, critters that are living in these different parts of the body. And they are fairly distinctively different in different parts of the body, which is interesting. Now, if you were around for a parasite show that I gave, there are things like the body lice and head lice and pubic lice. They're in those, the head lice stay on the head, the body lice stay in the torso area, and the pubic lice stay there. Anyway, so here's an interesting study. Somebody wondered, well, let's look at belly buttons. There is a long history of contemplating your navel, and so here we have, they take sterile swabs, rub it in somebody's belly button, and then streak it on agar plates, and you get, and so each one of these represents what was on a different person's navel. Now, each one of these these are probably the same thing. One of the things that you use to identify microorganisms, bacteria, is the colonial morphology. So each one of those represents a single cell that landed there and started growing. And these are actually resulting from fungal spores. Right. Now, I'm suspicious about this because they're so close to the edge that it's possible this represents a contamination of, because this happens if you're growing stuff in the lab in petri dishes. It's not uncommon for stuff to get under the cover and onto it. But anyway, it gives you an idea of what's going on. So they did this with hundreds and hundreds of people. And over looking at this, we see this pattern. And you'll notice, I don't know if you can see this, but there are the 4 major groupings of types of bacteria: the actinobacteria, Bacteroidetes, Firmicutes, and the Proteobacteria. And you can see the colors of these things represent the proportion found in that part of the body represented by particular organisms. And I think this is the Propionobacteria behind the ear. I don't know if you've ever noticed this, but I've noticed this about myself, that after a day, if I rub here, I can smell certain type of bacterium that makes a particular weird smell. And you may have noticed that if you don't shower much and you sweat, it'll start smelling. And that's because of bacteria that are living off of the stuff coming out of your body, out of the various pores in you. And they've done this in more detail. So, the summary here of parts of the body and what types of things are there can be seen here. And I'm going to show you a video once we've kind of gotten a feel for this. So, now the stomach, only 25 species typically found. The stomach is not really a very hospitable place. That we have cells in our stomach that secrete hydrochloric acid that is approximately pH 1 when it comes out of a cell. Of course, there's other liquids in there, so it gets diluted fairly quickly. But there's not a lot of things that are going to be able to spend a lot of time in the stomach. But there are lots of things in particularly the colon, the large intestine. So let's click on this. This should work, yes.
We humans are obsessed with cleanliness. We wear masks in public when we feel a cold coming on, we cover ourselves in scented soaps, and we are crazy about our hand sanitizer. No matter how much we steam, sterilize, or scrub ourselves, we're still outnumbered by our inner ecosystem. Meet your microbiome. The average adult human is made up of about 10 trillion cells. But how many bacteria do you think you carry? A billion? A trillion? How about 100 trillion microbes on our skin, hair, mouths, and inside of our intestines? You and I and everyone on Earth is mainly microbe. But we're not born that way. We start out in a sterile womb and we get our first dose of bacteria before we're even done being born. As we pass through the vaginal cavity, we ingest a tiny bit of this guy. It lives in our intestines, where we'll join about 600 other species that come along with our mother's breast milk. Their job? To break down sugars in that milk that we couldn't digest on our own. As we cuddle and kiss and play in the dirt, eventually over 1,000 species of bacteria will call our guts home. Early on, our little friends start fighting off bad bacteria and help teach our immune system the difference between good and bad. Mice without a microbiome are much sicker as adults, and children who take too many antibiotics, which kill good bacteria along with bad, can be more likely to get asthma and intestinal diseases later in life. These bacteria don't just protect us, they help feed us. Vitamins like B3, B6, B12, and K are all produced by our microbial friends and then taken up by our bodies. Our microbiome goes way beyond our guts. Anti-venom Leeuwenhoek became the first person to ever lay eyes on our little friends in 1683 when he scraped his teeth and placed it under a microscope. Even after we brush our teeth, hundreds of species live in our mouths, fighting disease and tooth decay. And no two people have exactly the same mouth bacteria. You share your mix with everyone you kiss. We used to think that sterile lungs were healthy, but there's bacteria there too, helping prevent asthma and lung infections. We've even found them in ears and arteries and manufacturing antibiotics in our noses. And then there's our largest organ, skin. About 1 trillion bacteria live in our hair follicles and epidermis. Some are playing defense, making our skin slightly acidic or producing chemicals to keep fungi from setting up shop. Some skin bacteria prefer to live in warm, dark places on the body, munching on skin oils and sweat and producing chemicals that give us a particular aroma. There's a lot of weird microbiome science that we don't understand. Regardless of your age, diet, gender, or where you're from, you probably have just one of 3 intestinal ecosystems. Like blood types, we all share only a handful of microbiome types across all people. Have you heard of bacterial mind control? The vagus nerve serves as a sort of direct phone line from your gut to your brain, and mice have certain friendly bacteria in their intestines are calmer, less anxious than those who don't. Here's another weird micro-mystery. If you looked at all the bacteria on your right hand, only about 1/5 of them would also be in your left hand. Even if you do this. All in all, we carry about 20,000 or so human genes and 3 million or so bacterial genes. In a sense, we're 1% human and 99% bacterial. Sure, some bacteria are bad, but most of them are just happy to call us home and happy to help us stay healthy. If we don't start treating our microbiome like the friends they are, they might rise up against us.
You are the 1%. So if anybody, said, I am the 1% in my world. So, this is a little aside. Carl Woese, really, I don't know why he didn't get a Nobel Prize, because some of the things that he discovered have absolutely revolutionized microbiology and the whole way we understand the world around us. It's phenomenal. What he realized back in the 70s, you have little bodies in your cells called ribosomes, and their job is to read messenger RNA, which has information from the DNA in your nuclei, and it reads it to make protein. Well, it's composed of several different little pieces of RNA, and these are relatively easy to isolate, but they have characteristics, particularly this one called the 16S, that refers to the size of it, this particular length piece of RNA. And it has some highly, highly conserved parts, things that essentially never change. And it has some other parts that are quite variable, because apparently they're not important which bases are there. And so it was the basis for his realizing that there are 3 major groups of organisms, 3 domains: the bacteria, the archaea, and then all the eukaryotic organisms, the eukarya. And these are based on looking at the 16S ribosomal RNA, certain parts of it. Now, the thing is that because it has its variable parts, you can also compare different species and see who's related to whom based on the parts of the RNA that are very variable. And in fact, a lot of the things that we know about what's where are not because we did the little swab and grew the things, because a lot of the bacteria won't grow in media, but we have identified them from their 16S rRNA. Because where does the ribosomal RNA come from? It comes from a living cell that is making protein. And if it's different than its neighboring RNA, it's a different species. So there are hundreds and hundreds of species that have been identified solely on the basis of their 16S ribosomal RNA. And that is part of how we understand the diversity of life on our bodies. So, our microbiome, this is actually would be the microbiota. I mentioned biota is the actual thing. So the mass of bacteria that is in your gut, obviously it makes up the majority of the mass of bacteria. And you can see different amounts of stuff, but you see that it's 3 pounds or so of material that is not you, that is on you and in you. So here is this might be a little difficult to see, and I will review this in a bit. So what are we seeing? We're seeing different colored bars based on different places. Blue is things isolated from stool samples. Cheek from the, swabs of the cheek, from plaque, from the tongue, from the nose, from the vagina, and from the skin. The length of the bar represents how much of it is, and whether or not, how many there are. And the darkness of the thing there tells you the density of the organisms. So this is the basic breakdown of things in these four major phyla of bacteria that I mentioned already: Actinobacteria, Bacteroides, Bacteroidetes, Firmicutes, and the Proteobacteria. So we're going to look at some examples of these things. Those pictures around the edge there give you a little bit of an idea of what's in each of those. By the way, this is the microbiome of a sponge, an ocean-dwelling sponge. So basically every animal in the world has its own little world, and different kinds have their own different things. So this is a hair. And they have stained it to show bacteria and fungi and stuff. So we have different components of the skin. You notice we've got different kinds of sweat glands that produce different kinds of substances, and therefore different kinds of bacteria will favor one type. And so we have sweat glands that produce different kinds of substances in different parts of the body. Like the ones in the armpit produce a different range of substances than the ones on the rest of the arm or on the forehead. And so you can see this would be the basis for why the body is like planet Earth, with different habitats all over it based on different amounts of moisture, different chemical foods for the bacteria and stuff like that. So we all have this little guy. This is one, the Demodex follicularum, lives in eyelash follicles. They're very, very small. I would show you one crawling around, but this is a link and not and our links we found are not working. So I'm not. They're so small that one could be walking across you right now and no one would know it unless they knew what to look for. You get a magnifying glass. Oh, there's a male one looking for a female to make that. Okay, so these are the major phyla or groups of bacteria. And each one of these is a very large cluster of different kinds of organisms. So we'll take a look at some of these. The actinobacteria, originally it was called the actinomyces, and some of them still have the myces part of the name. Mice means fungus. Before scientists or people that looked at stuff understood that fungi were one kind of thing and bacteria, prokaryotic organisms, were completely different, they noticed that these things grow as little filaments and they grow as little colonies. It looks like a fungal colony, but they're not fungi at all. They are bacteria. They're prokaryotic cells. Fungi are eukaryotic with a nucleus and stuff. So there are a number of things in this group, and in fact, they're very common soil organisms, and because there's a lot of things that live in the soil, they have for the past few billion years been manufacturing chemical weapons to fight one another. And in fact, a lot of our antibiotics are isolated from them or modified products from the actinobacteria. And streptomycin, you probably have heard of the antibiotic streptomycin, comes from that. Of course, the first antibiotic isolated and understood was penicillin, which actually does come from a fungus, penicillium. But these guys are not fungi. And you see there's a number of other interesting things that they make, all this kind of stuff. These are very potent antibiotics there. The Bacteroidetes These are very common in the gut. These are there's a wide number of these, and the genus Bacteroides is extreme. There's many species of that, and it constitutes a large amount of the flora, fauna of your gut. The Firmicutes. By the way, back up here, and I say GC ratio. When they first started looking at trying to make sense of the world, one of the things they noticed is the DNA, looking at the DNA, G plus C is guanine plus cytosine, so the ratio of guanine and cytosine to adenine and thymine was one thing. And so this is a group of organisms where they have a very high amount of guanine cytosine and lower amounts of adenine thymine. And this is a group which is characterized by having a low ratio. So basically all the critters in that one have a relatively low amount. Now, there's a lot of things in here that you're probably familiar with. This is Staphylococcus. Coccus means it's round. Staphylo means that they form clusters, that they divide in more than one plane, and so they form masses like that. Lactobacillus, these are all species of lactobacillus associated with the vagina. And that's what a bacillus means, rod-shaped, so this is a rod-shaped organism. This over here is Clostridium. Now, Clostridium, you may be aware that that is also the genus that contains the botulism organism, Clostridium botulinum, which has the most potent toxin that we've discovered. One thing about this is they're anaerobic and they will produce endospores, which are extremely resistant to most external conditions. This is why when you're canning material stuff, if you don't heat it to a high enough temperature to destroy those spores, then once the can cools off, they start growing in this anaerobic environment, and as they do that, they make the botulinum toxin, and when you eat it, you get botulism, and that's not so good for you. But proteobacteria, extremely diverse group of things, and it has 5 major groups. It's kind of like a fraternity sorority. They're the alpha, beta, gamma, delta, and epsilon fraternity sorority groups. And among some of them, the Escherichia coli, E. coli, coli referring, of course, to the colon. Escherichia coli, Vibrio. There are a number of different Vibrios, but one of them is Vibrio comma, which is the agent for cholera. And then Helicobacter. This is one that's very interesting because when you look at the prevalence in humans in this country, say 30 years ago, this was extremely common. Now it's relatively uncommon. But it also is the agent for stomach ulcers. Now the thing is that everybody had it, but not everybody has ulcers. So there's something else was causing damage to the mucus protection of the stomach, and then the Helicobacter would grow in there. But there's several species of that also. And one of the problems of widespread use of antibiotics has been decimation of certain kinds of things that were once very common in us. So again, our map of the diversity of things in us. Now there are a bunch of other, these are all the ones that are in low amounts. And interestingly, one of these is cyanobacteria. Cyanobacteria are also called blue-green algae. They're photosynthetic organisms. Okay, so just a couple of interesting members of some of these groups. This was the Propionibacterium. One I mentioned, there's several different species in there. These are the ones, some of them live right behind your ears. And propionic acid is kind of a sneaky acid, and some of them also make butyric acid, which is not that good. And then there are the ones that are responsible for acne and other skin conditions. They produce all kinds of interesting compounds. And if you like Swiss cheese, the bubbles, the holes in Swiss cheese are a result of these bacteria making carbon dioxide, which makes the holes in the Swiss cheese. Corynebacterium is these kind of club-shaped guys. One of them is the one that causes diphtheria. And by the way, it is diphtheria because that's a PH diphtheria. A lot of people like to say diphtheria. Well, that's fine. You can say whatever you want, but it's diphtheria. Streptococcus. You undoubtedly have heard of streptococcus. Somebody having a strep throat has got a particular species of streptococcus. And strepto means a chain, twisted chain. A coccus means a round, a spherical cell. And so there's some of them. And it says there are 50 species in that group. Okay, now this is interesting. Once upon a time, this genus, Prevotella, was given the name Bacteroides, which, as we saw, was a very common member of the gut. But they realized that it actually is a different species, and so they looked at gut bacteria of children in Burkina Faso, which is a country in Africa it's kind of near the Sahara, just below Algeria and those places. Anyway, and that bacteria made up 53% of the gut bacteria, and it is absent. Those were absent in European children of the same age. Interesting. And there's another one here that it says is possibly connected to rheumatoid arthritis. Rheumatoid arthritis is actually a autoimmune disease where your immune system is attacking the joints. What is the connection between those? I have no idea. I just saw this and that was interesting, so I put it in there. Don't ask me to explain it because I don't know. Now, this is one that you will hear a lot about, Clostridium difficile. Extremely common as a result of antibiotic treatments. Kills off the other bacteria, Clostridium difficile, then grows like mad. And we've got a video of it. A little discussion about
Have you
it.
ever wondered what it means to be human? Well, it turns out that only a tiny percentage of what you and I are made of is in fact.
So every time somebody talks about the microbiome, they go through the same spiel about how much it. So you're going to have to hear that. So if nothing else, you will know a little spiel of how to talk about the human microbiome that most of us think of ourselves about anyway.
Human. And we need these non-human bits to survive. But we're doing dreadful things to this hidden majority, and it's damaging our health in ways that we are only just discovering. It is said that we are 10 parts microbe and 1 part human.
There you go.
And that's counting microbial cells as the unit. If we count unique genes, then the difference is a factor of 100. We are only 1% human in terms of unique genes. So you and I are mostly bacteria of some kind. We need each other to stay healthy. And there's now a name for this part of ourselves. The human microbiome is a set of microbial communities that have co-evolved with us and we with them, and have become part of the human landscape. It is an extension of self. They provide to us untold functions that we don't have the means of executing. We're born deficient in our ability to digest certain kinds of foodstuffs. For example, the complex polysaccharides of plants. We can't manage those very well. Our microbes provide us the means of taking advantage. They also help us manage lots of other encounters with the environment. One of the things our microbiome does is give us an internal shield of friendly microbes which protect us against invasion by some of the nasties out there.
The good guys try to crowd out the bad ones.
It's competitive exclusion. Another term for it is colonization resistance. So, they man the beachhead and make it hard for others to arrive and find space.
So where are these microbes? Well, while most are in our bowel, they can in fact be anywhere where we're exposed to the outside world.
So we have them in our mouth. The mouth is a measly 10 million per gram. But it's only measly because in the colon we're talking about 100,000 times as many per gram.
So the microbes in our bowel, for example, help us to recirculate valuable nutrients and in fact assist us to keep our cholesterol down. So we need them and they need us. We do some terrible things to our microbiome, like assaulted with antibiotics.
Yeah, unfortunately for us and for them. Everything I'm going to tell you about the untoward effects of antibiotics has to be balanced against the obvious important benefits. But I think for too long we have felt as though you can't do much wrong with an antibiotic, so if in doubt, pull it out. And that clearly is not a good position to be taken. We've been asking volunteers to take an antibiotic even though they didn't really need it. We do it for short periods of time. We watch them before, we watch them after, and we're now doing second exposures a half year later. And the bottom line is, maybe not surprisingly, far more members of the microbial communities of the human body experience a severe impact. They're decimated, many more than we thought.
Now, in healthy people, who might only have an occasional course of antibiotics, the microbiome seems to recover quite well. That's not so true in people who are sick and weakened. As a side effect of antibiotics, they can become infected with a germ called Clostridium difficile, which causes hundreds of thousands of deaths worldwide.
So you're right, Clostridium difficile is the cause of large numbers of deaths, but even larger numbers of illnesses where there is a recovery, but after some cost.
One solution that some people suggest for this devastation to our microbiome is probiotics, substances which put back the friendly bacteria.
The evidence on probiotics is still early to interpret, but I would say it's suggestive that there is some potential benefit there.
As I said before, these friendly bugs, our microbiome, does a lot more than protect us against nasty infections. They keep our cholesterol down and detoxify poisons. So the question is, if we damage our microbiome with antibiotic overuse, are we causing heart disease and cancer?
Short answer is we don't know, because no one has looked for this particular link in the story. We simply don't know.
So while in the past we've seen bacteria as the enemy and got excited about how we can kill off disease-causing germs with antibiotics, it's time for a new way of thinking. Remember, we're talking about 99% of what we are. Have you ever wondered what it means to be human? Well, it turns out that only a tiny percentage of what you and I are made of.
So it moves too fast for me to catch it. Okay, so here is a little case study I found. So one of the things that happens in terms of creating a change in the microbiome is getting pregnant. So they did a study where they had some women that were pregnant and some that weren't, and they compared their microbiomes. And this is not an easy thing to do. There's a lot of controls, mathematical and statistical analyses that you have to do in order to actually make any sense out of this. Now, so you don't need to read this. It's just telling you what they did, and in general terms, the overall diversity and richness were reduced in pregnancy with the dominance of lactobacillus species and others, Clostridia ales, bacteroid ales, etc. And this was done by looking at our friend 16S ribosomal RNA. And this is, the green is the individuals that were sampled that were pregnant, and these are the blue are the ones that were not pregnant. As you can see, they do fall into two fairly distinct classes, and in general terms, the higher up you are here, the greater the diversity of the critters on them. Okay, now here's another situation that some people are not born in the usual way. In other words, they're born by cesarean section. So they do not pass. Now, in that earlier video, he talked about the birth, cavities. I've never heard of it talked that it's the birth canal, basically going from the uterus through the vagina and out. Anyway, regardless of that, there are people that aren't born that way. Now, in that time that you're getting pushed out, you're getting coated with bacteria. And if you're delivered by cesarean, then they cut open the tummy and pull the baby out, and it doesn't go through there, and so it gets a different set of bacteria on the outside. And is this something that makes any difference? Well, so what they did was they looked at babies that were just a couple of days old, and they got fecal samples and looked at the diversity looking at the 16S RNA. This is a gel showing basically a little bit of one organism and then some unknown something or other there. And this is the pattern what we see from babies that were born through birth canal. A much more diverse thing, just a couple of days after birth. So there is a distinct difference there. Now, what effect does that have? So this is a chart showing childhood diseases. These are factors relative to vaginally born children, the average for them. So you'll notice that in every case, it is higher than 1.0. 1.0 would mean it was exactly the same, indistinguishable. Anything less than 1 would mean that they had a lower incidence of those conditions. There's only one situation, type 1 diabetes, it ranges from almost no effect to a modest effect. But these are all different kinds of allergic rhinitis, in other words, getting hay fever and stuff, asthma, celiac disease, diabetes, gastroenteritis, having both gastroenteritis and asthma. So that difference in how you're born makes a difference in the organisms that are in you and makes a difference later in your health.
Have they figured out a way to compensate for that?
Yeah. Well, I guess you could get a moist rag, rub it somewhere, and then rub it on the baby. But that would work, I guess. I'm not sure anybody's tried that, but it seems not unreasonable. Yes.
What's the deal about repeat cesareans?
Some people was more than one birth by cesarean section.
What difference does that make to the baby? How does it know what?
Well, it does. What it makes a difference is in the frequency of that particular condition. This just shows you the results. What's going on there on another level is not evident from this data, but we see something is going on. It apparently does make. You might say, "Oh, it doesn't make any difference." Well, it doesn't make any difference to you, but apparently it makes a difference to them. Anyway, so everywhere in the world there are bacteria and stuff growing on things. Now we have biofilms in us too. In your mouth, on your teeth, every day you scrape off a biofilm called plaque. If you don't, eventually it forms this hardened thing called tartar. This is a community of microorganisms, bacteria, and that start wherever there's moisture, so in pipes, in tubing in hospitals, anywhere that there's moisture, there is going to be the possibility for the formation of a biofilm, a community of microorganisms. And they often have a very particular structure to them. So this is a surface. This could be, doesn't have to be your teeth. This could be in a pipe or in a catheter or anywhere that moisture is flowing and there are access to bacteria. They will, by electrostatic attraction, get stuck on the surface. They will start producing the slime. They will create this slime matrix. Other organisms will join that. They will actually develop channels in there that allow for flow of materials through it. This matrix of material makes the organisms that are in there highly resistant to antibiotics and to bleach. I mean, you can give a cursory bleach treatment to a biofilm, and you probably will not remove it, because that slime that they exude, and which they're all buried in, allows them to set up there. Now, there was somehow I missed something. Oh, here we go. I went by this really fast. I don't know. I guess because this is actually probably one of the most important slides, and I don't know how. I just went by. And then I said, gosh, where's that cool slide with all the two videos that are really cool? I don't know if you've ever seen these, but they have these things on the internet where somebody is talking and somebody is drawing something at the same time. And the first video is one of those about your gut microbiome. Sorry, I got this out of...
Throughout most of human history, we have felt at war with microbes. However, the human body is colonized by a vast number of microbial population, known as the human microbiome. Trillions of bacteria colonize our bodies. We can find them in various locations, such as our throat, our gut, and even our skin. In a healthy adult human body, there are approximately 10 times more bacteria than there are human cells.
How many more?
The microbiota, which is bacteria, have a symbiotic and mutualistic relationship, which means that us humans and the bacteria work together, work with each other, benefit of each other, and depend on each other for survival. There is a growing number of research into the field of the human microbiota. For example, the association between the gut microbiota and our health. It's an ongoing research, and new insight on the mutualistic symbiotic relationship are emerging rapidly. Most of these studies and research are based on in vitro studies as well as animal studies. Another important terminology we should learn is the microbiome, which essentially means a community of bacteria. So, for example, if I say the human microbiome, it means the community of microbes or bacteria that live within our oral cavity. In this video, we will investigate and focus on the bacteria that live in our gut. When we talk about the community of bacteria that reside in our gut, we can refer to them as the human gut microbiome. We will first learn about what they are and some interesting facts about them. And then we will look into the effects they have on the human body, which is, as I mentioned, an exciting and relatively new field of research. So we begin here with the digestive tract. Looking at the digestive tract, which is made up of the esophagus, stomach, small intestine, and large intestine, these microorganisms are mostly found in the large intestine. Though it is important to understand that we do find these bacteria also in the small intestine, just not as much. So let us recap the regions ascending colon, the transverse colon, the descending colon, Siegfried colon, and rectum. We also have the appendix that joins with the cecum. You might be thinking, though, if bacteria are inside our body, wouldn't it initiate some form of immune response? Well, that is true, but the digestive tract, as well as other tracts, are special in that it contains mucus, for example. So if we zoom into the large intestine here, we have the lumen of the large intestine. And here are the colon cells. Above the colon cells, we have mucus, which acts as a barrier between the microbial community within the lumen and the human cells. The highly dense microbial community and the host intestinal cell linings are separated by a thick layer of mucus in the large intestine. Interestingly, if we compare this to the small intestine and the microbes are only separated by a thin layer of mucus, much thinner compared to the mucus layer of the large intestine. Therefore, we can see how that mucus helps keep these bacteria from causing any harm. Also, the antibodies that are part of the mucosal system, as well as the tight junctions between the intestinal cells, help in this respect. So there is a lot of barrier here, allowing these bacteria to live within us in a symbiotic mutualistic relationship. So why do we have it? Why do we have these bacteria living in our gut? Well, the gut microbiota become established at birth and continue to change in composition throughout development. In fact, many factors influence the amount of microbes we have inside our body, such as taking medications, diet, and lifestyle. All are factors that influence the composition, the amount, and the different types of microbes we have. And therefore, each individual is unique in that each individual has a unique amount, composition of microbes. In the large intestine, the bacteria are mainly found in the proximal colon because this is where substrate availability is highest. But there are still so much bacteria everywhere else, such as the transverse colon as well as the descending colon. And so we can say that the bacterial density will increase from the duodenum of the small intestine to the large intestine. We find especially a lot of bacteria in the proximal colon. As well with this increase in density, there is also an increase in bacterial diversity. So there are more bacterial species as we move towards the end of the colon. With the increase in bacterial diversity and density, there is also an increase in mucus thickness. Now, the normal gut microbiota is dominated by anaerobic bacteria, but there are some aerobic and facultative anaerobic bacteria that are found in the intestine. In total, there are up to 1,000 species of bacteria living in our gut, but interestingly, only belong to a few known bacterial species. The most abundant bacteria come from the phyla Firmicutes and Bacteroidetes. Our gut microbiota play many fundamental roles in our body. Let us learn a bit about the effects the gut bacteria have on the host physiology. The main impact gut microbes have on host physiology is on metabolism. The gut microbiota has been shown to help absorb nutrients. But the main effect on metabolism is that the gut microbiota have a big role in fermentation. You see, foods that are not digested and absorbed in the small intestine, such as non-digestible carbohydrates, will reach the large intestine. And here, the carbohydrate will undergo fermentation by the bacteria. So let us have a closer look at this process. So here we have the colonocytes with mucus on top, and here is the blood supply. Within the lumen of the colon, bacteria communities, waiting for food to come. The non-digestible carbohydrate particles will reach the colon and will be then fermented by the bacteria. Through this fermentation process, the bacteria will produce a byproduct, a substance known as short-chain fatty acids. Some of these short-chain fatty acids, such as butyrate, are used as energy by the colon cells. Up to 70% of the energy used by colon cells come from short-chain fatty acids. Short-chain fatty acids that are absorbed into the blood can have other effects around the body. There is some evidence that short-chain fatty acids will influence adiposity by increasing lipogenesis. The short-chain fatty acids have also shown to influence the immune system in many ways, as well as they have been shown to play a key role in the health of colon cells. Another fundamental effect of gut microbiome is its effect on gut morphology, so gut structure. These effects include increasing the amount of mucus in the intestinal tract, decreasing intestinal cell permeability, deepening the crypts of the intestine, increase vascular flow, and stimulate tissue repair and renewal, particularly the villi. All of these effects essentially will promote barrier integrity, as well as the function of the digestive system. The reason as to how the gut microbiome modulates most of these activities remains elusive, but most of the research suggests that it is attributed to the production of the short-chain fatty acids following fermentation of non-digestible foods. The gut microbiota has also shown to have an effect on the immune system. For example, it has shown to influence the maturation and development of lymphoid tissues, such as Peyer's patches in the mucosal system, as well as the lymph nodes. The gut microbes are also able to regulate production of some immune mediators, including increasing concentration of secretory IgA antibodies. Recently, it has been found that the bacteria are able to influence the composition of T cells in the lamina propria. The gut microbiome also has effects on the nervous system. Most of these effects are based on animal studies and include decreasing synaptic connectivity and promoting anxiety-like behaviors as well as pain perception. There is also suggestions that the gut microbiota influences bone homeostasis by regulating bone density, as well as increasing the reabsorption of calcium from the gut. Now, we're still only scratching the surface of our understanding of gut microbes and how they affect our health. It is an ongoing and exciting field of research. Hope you enjoyed this video. Thank you for watching.
So, you may have heard of TED Talks. There's a related group of TEDx Talks, so that these are things that are not actually done at the place where the TED Talks are done. But there's one that was done at Caltech, and this is a video from that.
So it never ceases to amaze me that each of us carries around a 3-pound mass of cells in our heads that controls literally everything we do. Importantly, though, the brain doesn't act in isolation from the body, but rather it responds to the needs and experiences of each of our organ systems. Now, here's a staggering statistic that some of you may have heard before. Our bodies are comprised of 10 times more microbial cells than human eukaryotic cells. These microbes, which are primarily bacteria, but also viruses and protozoa, they're part of our normal flora, and they make up what's called the commensal microbiome. In the intestines, there are 100 trillion of these bugs, reflecting over 10,000 unique species, and contributing 150 times more genes than our own human genome. It's even estimated that collectively these microbes would weigh 2 to 6 pounds, which is up to twice the weight of an average adult human brain. More and more we're learning that these commensal microbes that make up us have co-evolved to play fundamental roles in normal brain development and function. So we can study the role of commensal microbes by raising mice as completely germ-free and recolonizing them with whichever microbes are of interest. And by these types of studies, we're learning that commensal microbes regulate several complex behaviors, like anxiety, learning and memory, appetite and satiety, among lots of other behaviors. So you can see now that by studying this microbrain interaction, we can learn really important lessons about how microbes can contribute or affect our brain health and disease. So you might be wondering, how in the world does a microbe that lives in your gut affect your brain? And there are many different mechanisms. One way is by activating the vagus nerve. So the vagus nerve contacts the gut lining and extends all the way up to the brainstem itself. And this is a mechanism by which the bacterium called Lactobacillus rhamnosus affects depressive-like behavior in mice. So in a task that measures depression-related despair, mice that have been treated with this bug exhibit less depression-like symptoms. And this is not seen if the vagus nerve is severed. Another way by which microbes can affect the brain is by activation of the immune system. So about 80% of the body's immune cells reside in the gut, and immune abnormalities contribute to several neurological disorders. This is one mechanism by which the bacterium Bacteroides fragilis prevents a mouse version of multiple sclerosis. So mice that have been treated with this bug are more resistant to the disease, as shown by the red line in this graph. And this depends on the activity of a special subset of immune cells called regulatory T cells that expresses the marker CD25. So if we block the activity of this immune cell, then the beneficial effects of the bugs are prevented. Another way by which bugs can affect the brain is by activating the gut endocrine system. So gut endocrine cells are primary producers of neuropeptides and neurotransmitters. Gut microbes themselves can also produce metabolites that could affect brain function. And this is one pathway that we think is involved in the microbe-based treatment that we in the Patterson and Mazmanian labs here at Caltech have used to treat autism-like symptoms in mice. So by treating mice with this bacterium, Bacteroides fragilis, we're able to correct core abnormalities, such as the communication deficit that's depicted here. That's a hallmark symptom or diagnostic symptom of autism. So here are mice that are autistic-like display less communication, as depicted by the blue bar, and treating them with the bug corrects this effect, as shown by the red bar. So I think that the implications of these discoveries is huge, because what if we could, without a single invasive procedure, treat disorders like autism, depression, and multiple sclerosis? Microbe-based therapeutics might offer a way to build a stable community structure that can impart long-lasting effects without the need for continuous treatment. Also, since microbes are relatively easy to manipulate and even eliminate, they can be readily modified for better functioning, regulatory control, targeting, and even delivery. So as a take-home message, I want you to remember that not only are we made up of mainly microbial cells, but that some of these cells can be truly mind-altering, affecting our brain development, function, and even our behavior. And also, in light of several studies showing important roles of commensal microbes in a variety of biological processes, from nutrition and immunity to now brain and behavior, consider all the things that we do on a day-to-day basis to change or disrupt our microbiome, and how this might affect our health and predisposition to disease. Thank you.
So we saw all this stuff. If this, the NIH, National Institutes of Health, has had this project, the Human Microbiome Project. Oh crap! It's a link. It's not going to work. Oh gosh, that's the problem. You get old, you just forget what happened 2 minutes ago. Okay, I agree to acceptable use policy. No, I'm not interested in expressing an opinion that you want to hear. So, Human Microbiome Project. This is for those of you that want to delve into this in more detail and get more of the technical kind of information. There are links to a number of articles that go into the details of how studies were done and what the results were. And there's all kinds of resources and stuff that you might find interesting. So, that's it.
Yeah, I was reading something recently, an article on this, basically on the same subject, and they were listing all of these things, all these diseases and stuff that are influenced by it. One of the things they talked about doing was fecal matter transplant.
Yeah, they do that.
And
in fact, remember the problem with the Clostridium difficile. For somebody that's chronically ill and has had to have antibiotics for extended periods, and they have gotten large amounts of the Clostridium difficile, basically replacing most of the others, they will get a sample from somebody that's deemed to be healthy, and now you don't. It's not a pill that you swallow. It actually is administered through the other end through a colonoscopy type of procedure. Because for one thing, if you swallow it, then a lot of those are going to get killed in the stomach. Anyway, so yeah, fecal transplants. It sounds so interesting, fecal transplant, but it is a fairly well-established procedure, and it seems, in fact, just 2 days ago there was a report of somebody that had got a transplant from his sister. He actually was quadriplegic, and so had all kinds of other problems, including this enormous Clostridium difficile infestation, and so that basically cured him of that particular problem. Didn't help the quadriplegia at all, but it did keep him free of that. In 2011, nearly 30,000 people died of Clostridium difficile, diarrhea induced by that. In this country, in the U.S. Worldwide, it's hundreds of thousands of people a year die from it.
That's more people in car accidents. Yeah, roughly the same. One of the things that they mentioned that kind of surprised them is that there was a case where there was a mother that had a weight problem and a thin daughter, and they did one of these things that suddenly the daughter began to have a weight problem. What they thought was incredibly kind of surprising.
Yeah. One of the things I mentioned early on was this is a relatively new field, and so over the next few years we're going to find out more and more about what's going on and how to use this to benefit people. Right now there are some applications, but a lot of it, is if you have mice that are autistic, then you've got a thing you can do for them. But in terms of applying that to humans, say they have mouse models of virtually all the human conditions that they're interested in studying. And they also have mice where they erase their immune system and put a human immune system into the mouse so that you can see kind of what it would look like in the human system. But it still is autism-like or multiple sclerosis-like conditions. These are all things that are somewhat analogous to what might be going on in humans, but they're trying it out in mice to find out as much as they can about it and see if there are some things that they might be able to then move into the human therapy realm. Yes, sir.
Years ago, there was this thought the appendix was a vestigial organ and blah, 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 but now is it true that they're thinking this is a place to colonize bacteria?
It is a place where you have a reservoir of stuff, yes, and so the colon can get repopulated to some extent after something happens, and then because that is kind of a little pocket that is a little out of the way, and so yes, that and also there's some things about the size of it and shape of it that even though it's very small and seems to be, vestigial, it's unlikely that evolution would get rid of it because it does seem to have a function. Now, in other animals, that part of the digestive tract that, so you have the small intestine comes into the large intestine, you have this part down here, the cecum, which is a blind alley. Well, in a rabbit, that cecum is giant, and it actually serves as a fermentation chamber where they do a lot of their breaking down of cellulose. And that's why rabbits have two kinds of poop, one that they re-eat, and the other that they leave around as little rabbit pellets. Because because that stuff is in the large intestine, it's past the small intestine where it can be absorbed as efficiently, so they re-eat that part. Go, Prophet G. Poop eating, yes.
I wonder if you know what we call broad spectrum antibiotics. Does they affect all four types that you illustrated here?
They would probably affect some members of all of them, but I don't know specifically. But the idea is to knock out as many different things as possible. But I don't know the details of it. And there's more than one type of broad spectrum antibiotics, so it's possible that there are ones that, if there's a doctor in the house, maybe we could find out if she knows anything about that. Broad spectrum antibiotics, which kinds of critters they kill.
So there's some that target gram positives and some that target gram negatives, and then the broader spectrum will target, some combination of both. So are you saying which one's a little
I'm wondering, from this, it's obviously not a good idea.
Yeah,
it's just a matter of convenience rather than culturing, seeing what the problem is. Let's kill whatever it is and everything along with it.
And the CDC and other organizations will have recommendations about don't start with these big guns, reserve them for certain conditions or certain situations. So hopefully your physician will practice by those guidelines. Well,
recently I went to India and I was given 2 courses, one for gastrointestinal stuff and one for respiratory stuff, as a general, guideline.
Were you sick or did they just give it to you?
No, in anticipation.
To take it if you got sick.
There's a gratuitous gross out slide. The defect action levels published by the USDA is how much stuff can be in your food that they.
We don't need to know
this
that.
is only if they're actually monitoring it, but pretty much they don't monitor anything anymore. It saves money. Guess what? If you don't do anything, it doesn't cost as much to not do it. Anyway, but these are the theoretical upper limits of how much stuff can be in your food.
So would those be good for our microbiomes?
Most of this stuff is, by the time you eat it, has been cooked. Yeah, so, insect fragments, those are very small, so the amount of extra protein or extra fiber that provides is probably minimal. But it's kind of cool to know that you're eating all this stuff, so that even the most hardcore vegan is actually eating some fair amount It's virtually impossible to eat anything without eating parts of insects and other things. And the chocolate, did it put chocolate on there? There's also the number of rodent hairs and rodent
peanut butter
turds that you can have per a certain amount. Anyway, but the one that I thought was really is mind-boggling. Hops, the bitter crap that the bitter material they put in beer to make it taste really bad.
Not an IPA kind of guy.
2,500 aphids per 10 grams. Do you know how much 10 grams is? It's this much. And so...
That's a joke.
It's not a joke.
Probably improves the taste.
Yeah, well, it seems like, boy, you get a lot of hops in there and you can get a lot of things hopping. Anyway, well, thanks for showing up and listening.